question was about
let's see, but I think it's better just to stop me at some point.
Stop you at some point.
Or just go on the 5 or 10 minutes.
Okay. So would you like me to kind of stand up and linger around here around 10:55, with about 5 minutes to go, or at 10 o'clock, at 11 o'clock? Yeah, we can do that. So I'll get up around 11 o'clock.
Yeah, you can see that I'm just flying over there.
Sure, sure.
It's better to stop.
Okay.
So what I should do is this.
Are you ready?
Yes, I think so. I can remember.
So in the interest of time, we're going to get started with our second talk of the morning, while some people might still be filtering in. So our second talk is going to be given by Joanna Sokowska on knots, lassos, links, and their origin, particularly in the context of proteins, following up on a talk that we heard earlier in this week. So thank you very much.
Thank you. Good morning, everybody. So this will be a little different type of talk. So I will try to tell you something about knots in proteins. So something on the border of, let's say, biology, chemistry, and physics, and with some kind of the modern application to the machine learning. It's difficult. So this is the overview of my talk. Do I have the pointer maybe? Somewhere that will be easier. Okay, so first I would like to give you some small short introduction about the protein. So it was Będlewo at the beginning of the conference. I hope so. She explained pretty well what is the protein. So probably you already know all the necessary information. Then I will tell you how we can define the knots in the proteins, and I will give you some example of proteins, and then I will try to move to present you different types of knots that can be found in protein. And if I have enough time, I will tell you about the different types of enhancement that can be found in the protein when you take into account also the side group, not just the main chain. It will work? Not really? Okay, it's okay. So this is the example of different nodes that you can see around in your daily life. So some nodes that can be used for sending. This is a very nice tree that you can see that's make links. Excuse me?
Is that real?
Yes, it's in Africa. So if you go for a nice conference, then you can catch something that you can use later on some presentation. This is some art that you can see. There's a different type of the knots, and the knots probably that you know there are knots from the physics. That was a Nobel Prize for this. This is something what we call in that you already know. This is the link, but it is now in the chemistry. It's a catenate. There was also the Nobel Prize for discovering such structure, and we know that DNA can form a knot. So where we can find the knots in the proteins. So just to remind you, this is a very basic information that it's just necessary for today about the proteins. So let's think about the protein that is composed of different types of the amino acids. What is important to remember that the amino acids, you can think about just a ball, let's say, or square, but some amino acids have the side group and they can make the connection. So there are 20 different amino acids. You read the protein from N terminal to C terminal. So you start from that side, go to this direction. So there is when you would like to define the knots in the protein, you know where to start. We usually start from the N terminal and finish with the C terminal. And for the later part of the talk, just remember that this amino acid can make the connection via the side group. And this is all what you need to know about the protein. I hope so for today. And some general description. So this is the protein. This amino acids, they can make a chain. This chain can fold. When it's folding, you can make different type of the secondary structure that we call this is the alpha helix. There are different type of the alpha helix. You can also make the beta sheets. The beta sheets are usually responsible for aggregation. This is when you have the amyloids. And the higher order is the three-dimensional tetride structure of the protein that you can see here. And they can make a complex, so they can connect each other and make the four-dimensional structure. So there was a question already asked in '94 by Mansfield. He asked if knotted protein can form a chain. And during this time, there was at least 400 proteins with no structure, so the structure that I showed you on the previous slide. And he scanned all this protein and tried to find the knotted protein. And his conclusion was there is no knotted protein. And he concluded that it's just too difficult for protein to make a knot. And at some point, this is what it was accepted for a very, very long time by biologists or biophysicists. So why is so difficult, or why the knotted protein was not discovered before? One of the reasons is that when you see this structure of the protein, then I can ask you, do you see, or could you tell me if this protein is knotted or unknotted? Maybe somebody would like to vote. Yes, that's his gambit. It's exactly like, yes, that's quite difficult, but it's your small kids, what you will find. You will find the termini of this protein. And what you will do, you probably will try to pull this, and when you pull, like a chain, then you can find out that indeed this protein possesses a knot. However, this is what we will call, then you can ask, we have some great topology people with the background with the knot theory here, and they will say, okay, this is not really a knotted protein or knotted chain because you can just untie this. Yes, that was kind of the untying. But if you will believe that you can connect this termini of the protein in infinity, so you can extend this to infinity and close without introduction of additional crossing, then you can find out that maybe we can call this as a probabilistic knot, and this protein could be knotted. So this is, I think Wanda gave a very nice introduction how to close the protein chain, but if you do this properly, let's say do this 1000 or 2000 closing on the sphere, then you can find out that there is some kind of the probability that will tell you that this protein is knotted. So we call knots in protein as a probabilistic knots. And today we can say that there are at least 8% of protein that are deposited in the PDB. So PDB is protein data bank, where you make the deposition of protein with known structure. So if you know the structure from some experimental data that I will tell you later on, then you can say that protein, if you know the structure, then you can just use, close the protein chain, then use a different polynomial and find out that the protein is not. However, if you will take into account as well the sides group when I told you before, so this is another example of the protein. So let's say this is the protein, and you think about the backbone. So if you take only this part of the protein without the sides group, then you think that this is unknotted structure. However, if you find out there is some strong connection, what we call the cysteine bridge, that is as strong to destroy as a backbone, then you can find out that this loop is closed. And then you can use, for example, the minimal surface approximation and find that one of the termini of the protein is causing this loop. So this type of the structure we call lasso. If you will make, if you find out that the protein can have two cysteine bridges, so two very strong connections, they are closing the red loop, and there is a blue loop that is also closed. And then you can find out, then you can find the minimal surface on red and find that there is the blue loop is causing the red loop. This is what we will call the deterministic link. And this type of the structure can also be found in the protein. And when you take more than one or two cysteine bridges in the protein, then you can find out that some of the protein make even with theta curves. And they can be quite complicated. So there can be additional knots on the chain and additionally closing, and this is the theta curve. So this is different type of the entanglement that can be found currently today in protein with no structure. So there is a kind of the fundamental question that people try to work on. So it's a more mathematical question, how to characterize entanglement in protein. So this is still, we can close the protein in the infinity and then find this probabilistic knot, but there is maybe other way to define the knots in the protein. There's biological question. As we know, there's only, so maybe I will say from the statistical physics point of view, we will expect that the long chain should be knotted. And from this point of view, you will expect that protein probably should have a higher number of knots, that plus 8%. Thus, if you take into account the protein with the preparing knot, this is not 8%, this is just 2% of no protein structure that possesses a knot. So this is really low number. And then you can think, okay, maybe because it's so difficult to fold them, as I will tell you later on, maybe evolution decided that it's too complicated to possess knot. But still, this 2%, and as I will show you later on, there in our body, there are some knotted proteins that are quite important for us. Maybe there is some biological role included in the knotted protein. How we can fold the knotted protein? So this is kind of the general question on the border of biophysics. Majority of the people, if we know how to fold normal protein, then you can ask how we can make a knot on the protein, and why evolution decide to keep or eliminate knotted protein. So we don't know yet still if evolution starts from random knotted chains that make a protein with a knot, or maybe this was a knotted protein chain. Yes. Okay, I think so. It's work. So then you can ask the question, maybe knots appear during the evolution. Maybe if we know now bigger number of knotted protein with secondary three-dimensional structure based on machine learning, maybe we will find out that there are higher number of knotted protein, and maybe we can track the evolution of knotted protein. So I will probably not have the time to talk about all different types of knots in the protein. So we constructed some databases, a server where you can find all proteins with knots. So this is the knot prot, and probably I will focus today on knot prot, but there is also the server where you can find the all classification of protein with last stop, links, and as I told you in the beginning, the chromosome can be knotted. So this is the database where you can find the, or you can scan chromosome to find if they are knotted. So what about, so it's a very big topic, the biological role of entanglement, but I will tell you a few things I believe what we know about knotted protein. So as you see, there is some advantage and disadvantage to possess a knot. So let's start with the things that worry us. We know they are prone to aggregate. During the folding, when you would like to make a knot, it seems that there's a lot of misfolding, and this could be the reason why not too many proteins are knotted, because then if there will be such huge problem with the folding, then probably we will not function as well. There are slower folders, so if you compare the protein with the knot and unknot, then you can find out that this one is folding, the time is longer, and based on, from the evolutionary point of view, it's better for protein to fold in fast way, because there will be not too much structure. There's many things going on in the cell, other protein is coming, so it's better that you just fold and conduct your biological function. Something that it's quite is good, so but it's not good for experimental investigation of the knotted protein. So if you have a knotted protein and you would like to unfold this protein and conduct some experiment, it seems that knots stay at the protein chain.
So if you would like to do the thermal denaturation or some chemical denaturation to investigate the folding, it seems that you cannot really untie the knotted protein. So if you cannot untie the knotted protein, you cannot really do too much from the experimental point of view. And because you cannot really, I did not talk about this, but there are some proteins that we know that are, they do not possess the native state, so they are in disorder conformation. Protein in disorder conformation you cannot crystallize because they do not have one stable conformation. So this protein with which is unfolded but with the knot inside the chain, you cannot really say if it was unknotted, where the knot is located. So due to this staying knots in the denaturated state, you cannot really investigate folding of knotted protein from the experimental point of view. And they are quite difficult of to be degraded. So once, when I was supposed to make kind of the experiment, we tried to denaturate the protein. We wait for 6 months. It was the experimental investigation. And after these 6 months, we find out that rather we destroy the protein. It was really decomposed, or there was a lot of misfolding, but we couldn't fold this back and we couldn't unfold fully. So that's kind of the, but there could be a positive way that protein, so going from this, we know that protein can survive harsh conditions. So some knotted protein can be found in hot springs, and they can be found in our stomach where you have also very atypical pH. So this is kind of the advantage for the protein. They can survive in harsh conditions. Something what I will talk later on, knots are very well conserved. So if you can have sequence of the proteins that are coming from the same family, we can find out that the knots is really well conserved. So we are all human beings here. We are all have some characteristics that are similar, and it's the same with the protein. It seems that the topology is the most strongly conserved characteristics of protein. So there could be a different sequence of the amino acids. They could have slightly different function, but it seems they all possess exactly the same type of the knot if you can classify them for higher order. We know it's quite difficult to show exactly the function of the knotted protein, but in the case of lasso topology, we can have the there's a possibility to show that given type of lasso is correlated with given function of the protein. So there is some kind of the correlation, and this is something what is very important. So some group of knotted protein are enzyme. Enzyme are protein that are responsible for they allow to catalyze the chemical reaction in faster way. And it seems that knotted protein, there are enzymes, they possess the active sites exactly where is the knot located. And there is a way to show that this catalysis is inside the knotted part of the protein, and maybe knot is responsible for formation of active sites that will facilitate conducting the chemical process. So, but what we don't know how, what is the evolution of knotted protein. So let me give you one more example why it's maybe interesting to study the knotted protein. So this is a very nice example of a protein that possess exactly the same biological function. They are responsible for some catalysis for methylation of anticodon loop in RNA. This is not important, but what is important, if this process will not take the place, the cell will not survive. So we need to, this is a crucial thing. And there are two proteins, one in bacteria that is responsible for this catalysis, and this protein is knotted. And the same function is conducted in our body by unknotted protein. So we have two proteins that are known from the biology. One is in our body, another one can be as well in our body, but this one is active, this one is knotted, and the active site is exactly embedded in the knot. So this we don't know why evolution creates such two protein with the same function but with different topology. But this can be done. This can be used. What is using now to develop new drugs. So this knotted protein is also pointed out as one of the is found in bacteria for which we need to find the new antibiotics. So what we are trying to do, and as well many other groups, we try to find the way to stop, to use the topology, so to characterize the active sites where we can just design some particle that it will stop only the bacterial protein, but it will not, it will recognize the active site that is knotted, and then we will find a way to stop the growth of this bacteria, but it will not affect our body. So the protein that is crucial for us. So let me tell you something about how we characterize knots in proteins. So that's this is something what we call the fingerprint. So to find where the knot is located on the chain, I already told you that we have this N terminal and it's going to the C terminal. And what you can do, you can just cut the amino acids from, let's say, from the N terminal, or cut the amino acids from the C terminal, and you can find out the minimal segment of the protein backbone where you will find a knot. And this is what we call, and what is quite important for another part of the talk, is a knot core. So it's not random position. We know exactly where the knot core is located in the protein. And this allows us additional very important information to find out. We need to find out if protein, I hope it will work because it's better, yeah. Hope so you can hear me. So let's assume this is the protein chain. And what we need to know if protein possesses a deep or shallow chain. And this is a shallow chain, shallow knotted protein. So if you have just few amino acids that you can cut from the N terminal, let's say, and you destroy the protein chain. And the same fact from C terminal. So this is the knots that are, let's say, are not interesting for us because they are shallow. And that means that they can unfold by the thermal fluctuation. Because we know that there's always some kind of the fluctuation in the cell, some temperature fluctuating. So if I have just thermal fluctuation, they are driving force to fold the protein in general, they can also be responsible for untying or tying the knot. So this is, we call this a shallow knot. If it's just 20 amino acids from one of the termini, that you can unfold this protein. However, there are proteins where you have something what we call a deep knot. So this is a deep knot that you have at least 20 amino acids from N terminal, and the thermal fluctuations are not sufficient to just by the random motion untie the chain. And it's the same about the C terminal. So if at least one of the both ends has to possess at least 20 amino acids to call this deeply knotted protein. So this is something important, and maybe I will show you some. Yeah, I will try to, yes. So and another crucial information, if you will take this matrix model, then you can recognize that if you so this is where you can see this is you have this knotted core of the protein. However, you can also try to find out if there are some internal knots in the protein. So to find the internal knots in or along the chain, not necessarily along the protein, you need to take subchain. And if you will take the subchain, then you can find out that some of the proteins can possess something what we will call the slip knot. And this is quite important. So just one more experiment. So this was your, let's say, your knotted protein, but one of the chain can just go back to the loop. And of course, if I will pull this, it will untie. However, this is the same way when you make your shoelaces. So when you tie your shoelaces, you make the slip knot conformation. So now you remember about tying shoelaces. That is how we make the slip knots in the protein. So if you just take into account part of the chain, then you can find out that if I crop this chain up to here and chain up to here, then I can find that the picture
on your right will show you that this protein possesses internal knot, and this internal knot we call the slip knot, and you can see by visual inspection this is the triple knot. So that's, I think, enough. So this matrix model is the way to characterize the knots in protein in more mathematical way. So we know where the knot is located. We know the size of N and C terminal, and we can find out if there are some internal knots that we will call slip knots. We have a different type of internal knots, not only just triple knot. So with this, we can I can tell you that there are protein. What is the most interesting? It was found, I think, already 4 years ago. There is a protein that is composed of 3 domains. So this is, let's say, the protein with 1 domain, but you can find out there are protein that is composed of 3 domains, 1, 2, and 3. And what you can see here that N terminal is just normal protein, but it's unknotted. The middle domain is knotted, and this one is also unknotted. So it's the most deepest knot that we know up to now, and then you can find out how this protein can make such a knot. What is important, there are this type of proteins we call the globular domain or globular protein, but there are also proteins that are embedded in the membrane. So this is the example of protein that is embedded in the membrane where you can find that it's as well possess a knot, just really, this is the trifold knot. And what is interesting, so we know that knots can be located in many different places. And recently we found, this is the picture of mitochondria. And why I'm showing this, because there are some proteins that are inside mitochondria, but they are not. So they have to be moved outside to the from this, let's say, blue part to the inside part of the mitochondria. So how you can move knotted protein here. And some knots can be done by accident during the experiment. So this is an example of the protein that was crystallized as a knotted one, because they would like to study this green part of the protein, this domain. However, it was unstable. So they connected they randomly by some linker, and they make just a knotted protein. And they find out that this conformation is very stable. One also important information. So this is the protein that possesses 5 knot. It can be found in your brain. So this is untangled. So if you have some problem with keeping the vertical direction when you drink too much, this is the protein that can maybe help you. And so it's abundant in our brain, and it also have the active sites that it's embedded inside the knot core. It's quite important protein because it's responsible for the degradation of other proteins. And why I'm pointing out this one? Because then you can find out that it's not just 5-2 knot. Then you can find out if you have this matrix model that there's also the 3-1 knot inside, 4-1 and 3-1. So that's the protein can, if you take into account all possible subchain, then you can find out that they are really complicated. So this is the 5-2 with the 3-1 and 5-2 protein. And what is interesting here, that this protein are coming from different organisms that are segregated by many years of evolution. However, they all possess exactly the same topological fingerprint. And we can show additionally that the sequence similarity, so you can compare the different amino acids between different proteins, it's very low, but the topology is conserved. So this is something what it will be used, or it's used currently with the machine learning to find out if protein possess a knot. So to conclude, knots are very well conserved in protein. So up to now, based on the structure from the PDB, we know there are at least 4 types of knots in protein: 3-1, 4-1, 5-2, and 6-1. And
what is important here that, so this is something that you probably know very well. This is the classification of nodes. And as you probably can see, there is some kind of the connection between this type of nodes that are found up to now in the protein. And this is important because this type of the nodes, they have unnoting number 1. That means that you can make this node just by cutting not in one place. And from the biological point of view, it's important, the last experiment, because you can make all these nodes in protein just by twisting. So you will have that loop, you twist this, and then you go with the dragging one of the chain across the loop once, then you make a trefoil node. If you do the 1.5, then this is the trefoil, the 4-1 node. And in the same way, you can make 5-2 and 6-1. So 6-1, as you probably know better than me, this will be the 6-1, just by pulling the terminal inside the loop. So it will lower your topological value. So that's quite important from the biological point of view. So then this is why maybe up to now it was believed that all knotted protein, or the protein that we find with knots, they possess the unknotting number 1. So the new one, or the protein that you could find, it will be the 5-2 type of the knot. However, let me just speed up a little more. So there is a different way how you can characterize, determine the structure of the protein. So you can do something what we call the NMR structure, NMR determination. This is for the protein that are more floppy, the proteins that are possessing the flexible structure. So you don't crystallize them, but you measure some NMR distribution of the peaks, and from this you can determine the structure. There's the classic way to characterize or to determine the three-dimensional structure of the protein is the X-ray approach. However, there's each 2 years there's something what we call the competition between the groups to try to predict the protein structure, and it's a very long kind of the competition. So every groups around the world can compete in this competition to predict the protein structure. So the structure is determined by the experiment. It's not published, and then all the theoreticians can try to predict the structure of the protein. And as you see that there's a kind of it was good, but not really good. And in 2016 and then 2018, people start to use the coevolution method and as well the machine learning method. And when they started to use this, there was kind of the breakthrough the last 2 years. So they, the DeepMind developed by Google company was able to somehow what we call the solve the protein folding problem. So they were able to predict the protein with almost 90% of resolution. So you have some score, I will not talk about this, that we call the PTT, that it's allowing you to compare the structure. The blue one is the structure that was known from the experiment, and the green one is something what was predicted. And as you see by visual inspection, I'm not showing the size group here, but the backbone is almost perfect. So currently the machine learning method are able really to predict protein with amazing quality. So what we can do with this? There's some, prediction is very good, but there's also the way to measure the prediction. So this is the one of the structure, the huge one that is predicted by DeepMind. And what is important to know is that you have the part of the protein that has a very high confidence to be predicted. So this is where you see this blue part. There are some things what is still accepted, that is the blue one. So the majority, the thought and general geometry of the protein is very well predicted. However, you have some parts that are more floppy, and you don't have the high confidence to show, to tell if they're really correctly predicted. But you should remember that some part of the protein are usually floppy. So protein has one native state to conduct the biological function, but as to conduct the biological function, you need to change the conformation. So if you're docking some other protein, you need to change a little. So this part has to be floppy to accommodate another part of the protein or accommodate the tRNA or different particles or binding some ions. So the parts that are responsible for binding the ions, they are usually outside of the protein. They has to be floppy. So this it's not high confidence to predict this, but it could be just floppy part of the protein. So generally, we use this PLDT score to tell you the confidence how well protein is predicted. So we apply the DeepMind, or we apply our algorithm to find the knots in the protein to all structure that was predicted by DeepMind. And we find out that a part of the protein with no knots, I should also show this one and this one, we find out there's, this is what we will expect. This will be the protein with the twist type of knot. However, we find the additional 4 structure of the protein that possess not twist type of the knot. So now you can ask the question, because AlphaFold was never trained based on protein with knots, so how he can predict the protein with non-trivial topology if there's only 2% of protein in PDB now with non-trivial topology? So there is no way to train this model. Additionally, there are no proteins with this type of the knot. So I could believe that the AlphaFold could predict the protein with no type of the knots, and there will be homological structure. But how AlphaFold could predict something that possesses different type of the knot, and there was nothing to train. So we try to do this by ourselves. So we also installed AlphaFold. We train AlphaFold by a structure that we know that should possess the non-trivial topology. And additionally, we check if all homological proteins, so the proteins that are coming from the same family, possess the same type of the knot. So we use the AlphaFold to predict. If we find that the protein is noted, then we find out all protein from yeast till bacteria and see if they all possess the same type of the knot. And we use it's not well known, but the Rosetta Fold is also the method that uses the machine learning approach, and it's also able to predict the protein structure. So in our approach, we use the DeepMind to predict all the structure. Then we make this, we check if knots is conserved in different organisms, and then we make the cross-validation. And based on this, we find out that this kind of the check can be survived only by 2 different types of knots. 5-1, there's a group of the protein with 5-1 knots that survive all our investigation, and as well the protein with double knot. So we can conclude there are new knotted family, but only 2 of those we believe they are currently predict in proper way. So something that it will be also interesting to tell you that based on AlphaFold, we can find out that there are some proteins that there is a crystal structure of the protein, and it was called before that it's unknotted. However, now based on the AlphaFold, when they predict the full sequence of the protein, then you can find out that this protein is knotted. So we need to rethink what is now deposited in the PDB. That's if it's saying that in PDB the protein is unknotted, that it's not necessarily true. Because in PDB, you only deposit the half of the chain, not a full chain of the protein, because this one will be too complicated for the X-ray structure to be determined. There's a different method to determine the structure, it's cryo-EM, and the resolution of this somehow is not always perfect. But now if you connect the cryo-EM and the prediction, then you can find out that maybe some of the protein would be knotted. So as I told you that it's only 2% of protein that are knotted, maybe this number is higher because there's the structure that are deposited in the PDB, they are rather small. and because they are just easier to determine the structure. So now if you use this both approach, then you can find out that some of the proteins can be really noted. And there is also something that you have to be careful. This is the example of protein that you can say, or we don't know if it's noted or unnoted, or maybe both options are possible. So this is the protein that was also known before in the crystal structure. It was on this part, but now they predict the full one, or we predict as well. But with the same confidence, we can predict that this protein will be unnoted, and the same confidence we have that this protein will be noted. And we try to also predict the homological structure, and this is 50-50. So we need the experimental verification to find out if this protein really is noted or unnoted, or maybe there are two possibilities, because this is also the part of the protein that is responsible for biological function. So maybe in some organisms, this protein will be unnoted, and in another one, it will be noted, but I don't have the experimental data for this one. And this is also example, I think, so Wanda talked about this. This is the protein with 6-3 node, and in this case, this is, as you probably can recognize, the node is conserved in human, mouse, and rat, but this is the something what we call the Danio fish that we cannot find out. So all the prediction from this organism is they possess 3-1 node. So you know that from 6-3 you can go to 3-1. But it seems that here we don't know if it's, maybe this is still mistake of AlphaFold prediction as well, our prediction that this protein possesses 6-3 node. Maybe it's just the 3-1. However, there is also something, what we find out that there are new type of nodes. So this is the protein with the double node. And in this case, we really try to find out if this protein is noted. So we try to predict the structure of all sequence possible. It was more than 500 sequences that we can find out that could possess the double node. We use this cross-validation. We use our methods to determine the topology. And what is important as well about this protein is a composition of two proteins that are deposited before in the PDB. So this is one protein that we know that this the single domain of this protein possesses a node, and this is another protein that also possesses a node. So we have the crystal structure of this one, and we know that they can form one long chain. And with this, we find out there is a little more of such structure. They are quite important. They are also found in some human pathogens. And because I also have the experimental lab, we try to determine the structure. And it seems that this is the crystal structure of first double knotted protein. So I think this is the final proof that protein possess non-twist type of the knot. And as you can see, this is the structure that we just determined, I think almost 2 weeks ago. It's a very good resolution, and it's also possess the two particles that have to be bound in the active sites, one and the second one, and they make a very nice double fusion knotted protein. And this will be probably the next one with the double knot. So it's not just one single protein family that possesses double knots. This one is a little more floppy, that it's a little more difficult for us to crystallize, but we hope that it will be soon deposited in PDB. And this is the proof that double knotted protein exists. So with this, then we can conclude that machine learning can predict the protein with non-twist type of knot, even though there was no proper training. And don't ask me how it's possible. That seems that something is above my understanding. Also, machine learning approach, usually with the machine learning, you don't need to fold the protein. So you solve the structure, but you don't know how to go there. So this will be, if I will have time, I will tell you something more about the folding. And this is the question, I leave as a question mark, because we still can predict the protein from the same family. They are possessed node and unknown. So maybe this is the problem with our training. Or maybe there are some family of protein that can be noted and unnoted in the same time, depending on the organism and maybe where this organism is located. So some of noted protein live in the harsh conditions. So maybe this protein that are in from organisms that need to live in harsh conditions, they will be noted to protect the protein against unfolding. But this is kind of my current hypothesis. I don't have the proof to show this. So if you would like, we make also the alpha node. So this is the database and the server where you can find out the protein that we check the topology, but you can as well use this server to check if your prediction will possess a node. And where we also, what is important, we use also this scoring method to tell you which part of the prediction is correct. So if you see that the node is located where you have the yellow part of the protein, maybe it's not a perfect prediction. But still, it could be correct because maybe this is the part of the protein that is flexible for due to the biological reason. So with this now, I would like to tell you something about the folding of the proteins. So what we believe, we believe that small protein can just, we call that they are minimally frustrated. So they can, by the thermal fluctuation and native contact, so the contact that exists in the native state, they can just lead the protein from the unfolded basin to the native one. And there is one single minimum that is the deepest one, but it's not necessarily the most important one because the protein is flexible, and the most stable conformation of the protein is not maybe this one that is necessary for the conducting biological function. So it's rather important that protein will fold inside this minimum, and in the case of the knotted protein, it has to be probably not the deepest one where the protein is the most stable. However, then you can ask the question, this is what it's called the funnel landscape, and then you can ask the question if native contact are sufficient to fold the protein with non-trivial topology. So this is something what we explored already long time ago when I was PhD. Still, so it was last part of my PhD thesis. So we take one of the protein, it was one of the first one was discovered. This is rather small protein with node. However, as you see, it's a rather deep node, so at least 60 amino acids from both sides. And we find out that based on just simple vanilla model, so when you have this native interaction, so the interactions that are responsible for stabilization of the protein in the native states are sufficient to drive protein from this unfolded conformation to the native state. However, as you can see, the success rate is very low, but it's still kind of the proof that you can do it. And what is critical here to understand is that you fold via the slip knot conformation. So you don't drag the terminal to fold the protein, or it's what we believe up to now, you need to make the squeeze loop that is already located in the native state. And then to minimize the energy barrier, you don't try to, if you close your eyes, it's quite difficult to find this loop. So to minimize this entropy cost, you make the slip knot conformation. So this is how you tie your shoelaces. You start to have some kind of the native interaction here, and then you slowly, by the thermal fluctuation, make the slip knot, and then by the thermal fluctuation, you drag the terminal on the other sides. And this is how you can also recognize this is what you will call, this is the Rademeisner move, the first tree one. So this is how we explain the folding of simple protein with native node. Yes. And then we use exactly the same kind of the method, so the coarse grain method, to fold the protein with 6-2 node. So you can twist, and then drag one of the terminals. However, now you can ask if we know currently that protein with the such type of node exists, and we have the double fusion, how we can fold such protein. So today I can probably tell you only maybe, or maybe not something about folding this one, but I can only have the prediction maybe how we can solve the folding problem here. So just to tell you, this is the example of the two experimental paper that they were able to show that it's possible to observe or to detect the protein can fold in pre-cell systems, so without any additional help. However, if you attach some additional domain that you can see that are similar to this, what we find out, the deepest noted protein, that maybe this protein is also possible to fold without any additional help. So how many minutes I have? I need to plan this correctly.
In about 4, I think I'd like to.
Okay, so this is the example of how you can think about the folding of the protein. So you can have this, what it was believed up to now, or I still believe that some protein can fold in that way. So you make the native twisted loop, and then one of the termini can drag, can be pushed across this loop, and then you can fold the protein. You can think about the things that it was that is the most common one in our daily life, about random knotting, or this is something what we are currently suggesting about the folding the protein with the on the ribosome. And to make this shorter, we find out that it's not possible to fold this protein via this pathway, but if you cut this, the N-terminal domain and C-terminal domain, and you have only the green one, we find out that it's possible to fold this protein. And as agree with the previous data that you make this secret conformation, we find out that it's impossible to fold the protein with the random knotting, because you can make a shallow knot, but the shallow knot will never proceed to the deep knot. So this is no way that you can fold such protein. And we find out that maybe it's possible to fold via ribosome, but I will skip this. So this is the ribosome, and you can present the ribosome in a rather simple tube model. And if it's possible, I will show you the movie. I hope so it will work. So we have a fun doing a lot of movie in my group. So that's I think this is this one. We need to skip this. So we start with the chain is moving out of the ribosome, and this is how the chain is pushed by production of the amino acids. And the green part is the part that we are expecting to see the knot. And as you see, we are not doing anything. We are just observing moving the chain outside of the ribosome. And what we observe now, we observe this normal formation of the native contact between some electrostatic interaction between the ribosome surface and the protein. And what you can observe here, it was the native twisted loop formation that was just dragged by the thermal calculation. And now what you observe that this nesting chain is just pushed outside of the ribosome. So this is the way how we believe, and because there was just normal native interaction, that it can diffuse outside of the ribosome. It's a very simple model, but this model allows us to explain folding in the ribosome. So maybe just to stop this before we will go further. So I believe that this is maybe the way how protein can fold, and I believe that maybe this double knotted protein on one side can be folded, the knots can be made by the ribosome, and the other side this will be the slip knotting mechanism. And maybe with this I will finish and only just show you the. Yeah, I have too many slides. Yeah, so I will finish with the group presentation because it's necessary to get the credit for that. Yes, so this is the majority of the people that were involved in this research. So Hanna, you know already, she came to my group a long time ago and she's still there. and I'm very happy about this. And the people responsible for the server development, and as well, Ola Yarmoliska, that it's currently in Google. So that's, I lost her, but she defended her PhD, and this was a normal life. Thank you.
We have time for a few questions for coffee.
So in this, well, first of all, thanks for the wonderful talk. And in this very first sample that you mentioned, about 400 proteins, and only one showed up to be a knot. This is less than the 2 or the 6%. Is this only due that they by then had only the chance to look at simple examples, or later on perhaps others turned out to be knotted as well?
I think there will be other one that will be not, but we need to explore this a little deeper. I will say I believe that if we can now crystallize the protein with double knot, that is showing us that the prediction is correct with the machine learning approach. Doesn't matter if we use the AlphaFold or Zeta fold prediction, and the structure that are deposited in the PDB, they are rather short. So I believe that we could expect more double knotted protein, if I understood your question correctly.
No, I mean on this very early slide, not your work, but
yes, from Mansfield. From
yes.
So at that time there was one protein that was knotted, but it was with the shallow knot, and he mentioned this one that it was with shallow knot.
No, I just wondered because there were 400 things he analyzed, and only one turned out to be a knot. So either he missed a lot of knots, or the kind of sample was so that there weren't knots, right?
Yeah. So the second your answer. So it was just at that time they crystallized the structure with the different families, and this one that he found it was noted it was carbonic anhydrase. But the other one was just not from this family that we know currently.
Other question for you, Anna?
Thank you for your talk. So I'm interested about the data representations of what is the output of the AlphaFold algorithm, and how can you compare it to the real experimental data to validate that the protein folded in this three-dimensional structure like correctly?
So there's at least 3 main outputs from the AlphaFold. So from the AlphaFold, what you have to give for the machine learning, you need to get something what we call MSA, multiple sequence alignment. So it's alignment of at least, I think the smallest one is at least 100 amino acids that you align. And this is the first input. The second one, you can give the structure that you believe that it is the homological structure, the 3-dimensional homological structure. And yeah, this is 2 input file. And in the end, you can as well obtain the similar output. So you will, AlphaFold will make the new alignment based on your alignment. So the one of the output is the new MSA. Second one, you can obtain the structure. And usually from AlphaFold, you have at least 5 different predictions. And if you know that there is an homological structure, then you can make the superposition and calculate RMSD between these 2 structures, or GTT, or any other kind of the superposition to tell you which part of the protein is similar to what you know. But there is still a lot of protein that it's impossible to find the homological structure or something that will have enough sequence homology to tell you that you make the superposition. So we believe that something that is lower than 30% of sequence similarity is not sufficient to make a judgment that this will be the homological structure.
So in the interest of time, I propose that we hold our additional questions for the coffee break. So please join me again in thanking both of our morning speakers. We'll start with the assembly plenary talks at 11:35.
Yeah, that's thinking about the hour.
Okay, 11:35.
That was a great talk. I have.
Yeah,
There's a line that
I
point.
should talk to everyone.
Why do you think that
that
when alcohol predicts these two types of thoughts,
Oh, of course.
that it's the
I'm making
alcohol
sure.
that's somehow right and the crystallographers were wrong and not the other way around?